Okay, after all is said and done, after we figured out what the different cellular organelles should be, I didn't want to grade it, but I also didn't want them to feel that there was no evaluation for what they created. So, I created a table area where my master scientists, the ones that were, typically are on top of their science job, at least a few of them, and I said, I want you to place each of the created cells, the animal cells and the plant cells, into their respective category. And so these are the results. Some people created standard animal cells. Others had mutations. And others were extremely mutated animal cells who had compromised viability. Okay, based on the organelles that we standardized. Now, nobody who created a mutated plant cell went all the way to extremely mutated. So at least the plant cells were just a slight mutation, which means evolutionary. They might have had an evolutionary difference, but they weren't exactly, like catastrophic, monstrous looking. And these are like the standard plant cells based on the organelles. Now, what we mean by extremely mutated is, say, for example, we called these rough endoplasmic reticulum pieces, but we've got locomotors at the end of that. And so, to me, that's kind of like, wait, what happened? I mean, we do have the DNA on top of the nucleus in the center. We do have one mitochondrion. That's a concern for me, because you have so much locomotors, yet you only have one energy cell in your cell. Okay, you only have one energy provider, one ATP producer. So over here, this one doesn't even have any mitochondrion. Oh, it does. But the mitochondrion are here, and you've got Golgi apparatus attached to the mitochondrion, and those are supposed to be protein makers. Why are they glued to the mitochondrion? That's not normal. Okay. And then the endoplasmic reticulum, this is rough, this is smooth, but why do they have smooth endoplasmic reticulum surface floating around freely in the cytoplasm? That's a strange thing. And this endoplasmic reticulum is all over the place. Okay? I mean, it's like, these are actually, we called purple disease particles because we didn't figure out what to do with them, so those are diseased particles. And so some people have decided to leave the diseased portions in there, even though we went over it over and over again. So, those are extremely mutated. They've got viability compromised, but the bottom line was, did they learn what the organelles meant, what they were for? Now, we take a look at this. And I don't see, oh, this one's a mutated animal cell that's able to exist, but there's no Golgi apparatus on it. How is that possible? Okay, Golgi apparatus would be the yellow. It has a little docking station on it. Okay, now this one's interesting. What's questionable? What's questionable is they're using endoplasmic reticulum pieces, and they have and they have like locomotors on the ends, but it's not so bad. And it's like maybe 1 point of mutation as opposed to these, which have like several points of mutation. Okay. And so over here we've got some mutated animal cells, slight mutations. Over here, these are all standard because they all have the Golgi bodies or the Golgi apparatus. They have some cell membranes that have nutrients in the receptor cells. We've got vacuoles. Okay, and in an animal cell, you typically should have one vacuole. You could have two, but typically, an animal cell has less vacuoles than a plant cell. This one, at first glance, looked kind of odd. Then I looked at it later. It actually had 3 mitochondria. Now they reduced it down to 2, which didn't make sense to me. Because if you had 3 mitochondria, you could support this much locomotion. These cilia and flagellates over here, these are the microtubules. And we allowed them to put a steering wheel on the nucleus because, besides DNA on the nucleus, the pink square, the nucleus does steer the direction of the cell and what it's supposed to do. Now we'll take a look at some of these plant cells here. And the plant cells, you know, you've got the more vacuoles typically, so you have at least 2 vacuoles, as opposed to the animal cells, which only had 1 vacuole each. And you got the smooth and rough endoplasmic reticulum, and these actually are the ratcheting pieces. I like those. Microtubules. These are ribosomes, little tiny pieces free-floating all around the cell. And then we have, the difference is chloroplasts. Now, we didn't have anything that could actually be called cell walls. You just assume the cell walls, or these are cell walls, but these are also interchangeable with cell membranes. I haven't found anything that really I could call cell walls yet, but maybe in the future. We could actually put it on the bottom. Maybe next time I go to Lego store, we'll figure that out. But yeah, and then we have slightly mutated plant cells. Now, what are the mutations here? This one actually looks pretty good to me. I don't see a mutation here, unless there's missing something. Oh, these are ribosomes. No. Those are the little ones. Those are ribosomes. These are lysosomes. They have docking stations because they accept nutrients, and they accept ribosomes on this position here, or on this docking station, because it has it recycles old cell parts, but it also processes nutrients. This one doesn't look like it's mutated. This one should go here, unless I'm missing something. Looks good to me. Okay, this one over here, maybe it's because it only has 1 chloroplast. The green is a big difference, that 1 chloroplast. This actually looks like it's a standard one. I think, oh, I know why. Because when I moved things to the mutated plant cell position, some kids were coming back and saying, oh, let me fix it up. And so they were trying to fix it up to make it more less mutated looking or whatever. That's probably what had happened. Over here, typically I prefer smooth, a smooth single endoplasmic reticulum, and then another rough, but, you know, this one is kind of combined. And they have a locomotor on a plant cell. They have cilia and flagellates on a plant cell, which doesn't make sense, because I thought plant cells don't move. They just, like, if it's algae, it's just in the water, and the water moves it, and it just floats around in water. That's different, because that's why and it's got 2 mitochondria, whereas plant cells only require 1 because they require less mitochondria and less energy, since all they're doing is floating around. So this is extreme. I call that extremely mutated plant cell. That's a compromised viability one. But it does have 2 mitochondria. I think it's just completely combined. But it does, it has the chloroplast. We'll put it in mutated. It's just a plant that walks on its own.